Okay, well, good morning, everyone, and welcome to the third lecture in the series, Innovation Gainesville. Today's speaker is Dr. Todd Brusco, and he will be talking to us today on how our immune system develops and how genetic variability and susceptibilities contribute to the development of autoimmune type 1 diabetes, which was formerly referred to as juvenile diabetes, and how age and environmental exposures affect aging of our immune system. Dr. Brusco completed his Ph.D. at UF's College of Medicine's IDP program in biomedical sciences and conducted his postdoctoral fellowship at the University of California in San Francisco, where his work contributed to the first clinical trial employing regulatory T cells as a potential treatment for type 1 diabetes. Following his postdoctoral fellowship, Dr. Brusco joined the University of Florida faculty in 2010 and subsequently joined the senior leadership team of the UF Diabetes Institute in 2020, where he is the principal investigator with nearly $11 million in direct extramural funding to study cellular engineering and genotype-phenotype studies in type 1 diabetes. The research aims of Dr. Brusco's lab revolve primarily around understanding the mechanisms by which the immune system maintains a state of control, referred to as tolerance, and understanding how genetic risk variants and age influence this process, as well as identifying pathway defects in individuals who develop autoimmune diseases. Dr. Brusco currently works with Dr. Mark Atkinson at the Sid Martin Biotech Complex at OneVax, which is a preclinical stage biotechnology company committed to developing novel and effective therapies for type 1 diabetes and other autoimmune diseases. OneVax has developed a proprietary biomaterial vaccine delivery platform consisting of subcutaneously injectable microparticles that encapsulate specific immunomodulatory agents capable of restoring immune tolerance designed to ultimately cure type 1 diabetes. So with that, I'd like to welcome Dr. Todd Brusco, whose talk today is on the human immune system development in health and disease. Dr. Brusco.
Good morning, everyone. Thank you for having me. And hopefully this remains very much an interactive presentation. So if you have questions that arise while I'm talking about this, immunology is one of these areas that is a fascinating topic, and obviously for what's been occurring recently has gained incredible interest in the field of COVID research and trying to understand why some people are better responders to COVID and others not. But I've spent at least a decade of my career as an independent investigator trying to understand what goes wrong in the immune system in individuals that develop conditions like type 1 diabetes, which is an autoimmune condition. So it's very different from type 2 diabetes, where you have a resistance to insulin and can be modified by diet and lifestyle changes. Type 1 diabetes very much is genetically dominated, and it results from the immune system attacking the cells that actually make insulin. So individuals that have type 1 diabetes have to take insulin shots and glucose monitoring throughout the course of every day. So very different diseases, and type 1 is primarily what we refer to as autoimmune in nature, so caused by cells of the adaptive immune system. So I'll give a couple highlights that are emerging out of the T1D Institute here, and I'll talk about some of the impact of age in the immune system as well. So as I mentioned, and if you overheard the conversation starting off, type 1 diabetes is incredibly heterogeneous, meaning that it can start in babies. We see the first signs of autoimmunity, these autoantibodies that recognize insulin and some of the other molecules in beta cells as early as a year of life. But individuals can develop type 1 diabetes throughout the entire course of their lifespan. So the first sort of hints that this was autoimmune in nature in addition to those autoantibodies that we find in circulation, is if you actually look within the pancreas itself. So this blue region that you can see here, can you see my mouse on the screen? I'm not sure if it's showing up. Anyway,
Yes, yeah, it's good.
Good. Yeah, so these green dots and these red dots are T cells of the immune system that are now recognizing insulin producing beta cells as foreign and causing their attack and death of these targeted beta cells. So we see direct infiltration of the pancreas of cells of the immune system that are responsible really primarily for killing off these insulin producing beta cells. So when you look, as I mentioned, there's a lot of variation in who can develop this disease. So here, these are large numbers using a survey that was conducted by a charitable foundation called the T1D Exchange. And you can kind of see the ages in which most individuals develop disease. So clearly, it occurs most often in young individuals. So some of you may have grandkids of these ages. But typically, we in our pediatric clinics here, we see disease peak around 10 to 12 years of age locally here. But it can occur across the spectrum of ages. The other key thing that we're finding out is that when we use drugs to intervene in the immune system, and one of those drugs is this molecule that we refer to as anti-thymocyte globulin, which is a combination of antibodies that targets primarily T cells of the immune system. We find that some individuals are very good responders to this drug and others don't respond at all. So a lot of what we're doing in the lab now is trying to understand why some individuals are better responders to drugs like ATG and others simply fail to respond, so that we can tailor treatments that will more effectively treat individuals so that we avoid side effects in individuals as well that are not going to respond. All right, so I mentioned already that type 1 diabetes is autoimmune in nature, and we've conducted genetic studies here at the University of Florida, and in collaboration with collaborators throughout the world, we've built up a long list of genes that are associated with risk for disease. So there's now almost 90 different genes throughout the entire genome that confer risk for type 1 diabetes, the largest of which is this molecule called HLA, or human lymphocyte antigen. So this is one of the genes that your body uses to present things to T cells in the adaptive immune system. And it's also one of the reasons why if you ever get an organ transplantation, you have to kind of match these HLA genes so that the organ is not recognized as foreign and rejected. But this is this single genetic locus within the genome is responsible for over 50% of the overall risk for developing type 1 diabetes. So almost 90% of the people that have type 1 diabetes have either DR3 or DR4 genetic risk elements. And what we can now do that is actually very helpful and powerful is that these genetics are incredibly complex, but we can now take the individual risk at all of these different loci throughout the genome, and we type individuals at almost a million of these single nucleotide polymorphisms or slight variations throughout their genome. And we can combine all of that genetic information into what we refer to as a genetic risk score, a simple number that tells essentially an individual how much risk they have of developing a complex disease like type 1 diabetes. And this becomes incredibly powerful at a population level, because what it allows us to do is really identify individuals who may go on to develop disease before they ever develop symptoms. So what happens currently is that if someone develops type 1 diabetes, they essentially go go into metabolic crisis, and they may wind up going into the hospital emergency room. As a young child, it's very confusing. Oftentimes, the parents will think that they have a very severe infection or something like that. So they'll go in and they'll often be in diabetic ketoacidosis and be very, very sick. So we've pushed a lot of effort into understanding the genetic risk of this disease so that we can type, hopefully, individuals even at birth and understand perhaps how much risk they have for developing these complex diseases like type 1 diabetes. All right. So I mentioned already that my laboratory has a strong interest in understanding how the immune system changes over time. And one of the clearest examples of this change that we see over time is that as we age, all of our T cells come out of a small organ referred to as the thymus. It's the site of origin of T cells, and they come out, and we can see this here from an individual. This is umbilical cord blood, and each one of these dots represents a cell. And we're looking at hundreds of thousands, if not millions, of cells that is creating this plot that you can see here, representing all naive cells. So our immune system comes out and it's a clean slate. And most of those T cells are naive and they're ready to respond to all sorts of different infections. This is, I think, one of the major reasons that young kids actually do quite well if exposed to COVID. And then as we age, our immune system changes. It moves from what we refer to as a naive T cell state into what we refer to as a memory T cell state. And you can see that change in what these two different plots look like here as people acquire T cell memory over time. So if you think about, early life and development and your kids, they've gone through an incredible number of challenges. They've gone through exposures in daycare. They've received lots of vaccinations, and all of those influences throughout your life change your immune system and help you develop immunological memory. So what you're looking at the bottom here are trends in what these cells look like to about the first 2 decades of life is where we see the most changes in the immune system over time. So we see a loss of or a reduction in naive T cells, and then an acquisition of immunological memory, or here shown by CD4 T cells, increasing over time. And we can see that for both a type of T cell called a CD4 T cell, as well as CD8 T cells that fight off things like viral infections. So what that essentially inspired us to do is to try and build what we refer to as growth charts for the adaptive immune system. So for many of you that have your own children, you may recall taking them to the pediatrician office and having their heights and weights plotted out, and the physician saying, I have 4 children myself, so I'm well aware of this process of taking kids into the clinic and having their height and weight measured and tracked over time. So we have a great idea of where kids should be in terms of these simple metrics of height and weight, but what we lack is a very basic understanding of what your immune system should look like over time. So my laboratory and that of my collaborators here at the University of Florida Diabetes Institute have collected thousands of samples from patients and controls. And we're now beginning to sort of build out this idea of creating growth charts for the immune system. So we look at 150 different types of cells that we can find in a small amount of peripheral blood. So you can think of a couple drops of blood essentially providing us with an idea of how the immune system is changing over time. And we're now building these models. And one of the interesting findings of this effort, really going back, looking at what changes over time, is that we can make a very accurate prediction of how old someone is with just looking at their immune system. So we can have a very highly accurate prediction that someone, say, may be 46 years of age, just by taking a drop of blood and understanding what their immune system looks like. It's a very set program at how this changes over time. All right, so moving forward. So much of what we do involves interacting with patients in the clinic. We also run, and I think you've heard talks by my mentor, Mark Atkinson, who really developed this program that we refer to as NPOD, which stands for the National Pancreatic Organ Donor Network. And the idea was that the field has spent something like 3 decades trying to understand type 1 diabetes, which is a disease of the pancreas, by looking in peripheral blood. And we struggled for decades, really, to try and understand what's happening. And we came to the very simple conclusion that we were never really going to understand this disease unless we were able to actually look at the pancreas itself. So we set up this whole nationwide organ donor network, where if anyone happened to pass away across the US for various reasons, sometimes it's because of, unfortunately, multiple or motor vehicle accidents or other reasons, that if they had bio or markers of diabetes, that we would want to acquire their pancreas if it didn't qualify for transplantation. So we've done that now from over 500 individuals at the University of Florida, and we provide sample to researchers throughout the world. So over 200 affiliate researchers throughout the world to really begin to understand what is causing the disease at the target organ itself. So it's really, I think, transformed the landscape of trying to understand this disease process, and also how to really intervene in the whole process as well. And here's just showing some of, it's not just individuals with type 1 diabetes. We get cases from individuals with type 2 diabetes, as well as various other reasons. Some individuals that have survived type 1 diabetes for over 50 years and eventually wind up providing their organs for research purposes. So one of the key sort of observations of this program is that in the past, there's an animal model of type 1 diabetes where we use a mouse called the NOD, or non-obese diabetic mouse model, that has some similar features to the type 1 diabetes that we observe in humans, but varies quite dramatically. So it has lots of, each one of these blue dots down here that you can see on this tissue image represents one of those immune cells that is infiltrated into the pancreas and is destroying these insulin producing beta cells. So when we look in the pancreas of humans with type 1 diabetes, the amount of lymphocytic infiltration is oftentimes much reduced compared to what we see in mice. So what we can sort of gather from this is that it's a much more heterogeneous process in people, as one might expect. So this mouse is all genetically identical. Ultimately, it kind of represents an N of 1 in terms of numbers. So we get a much better picture of the disease process by actually looking, not surprisingly, at individuals that carry the disease itself. So one of the interesting observations of the NPOD program, this is actually images of the pancreas of an individual on top that has type 1 diabetes, and an age and weight matched individual on the bottom that would represent a normal, healthy control. So the pancreas, as you can kind of see, has this head region that is slightly larger, and then it moves all the way down to the tail. But one of the key simple observations that we've made through the MPOD program is that the overall pancreas is as much as 40 to 50% reduced in mass in individuals. So overall, it shrinks essentially in individuals that develop type 1 diabetes. And what we've actually found now is that even before someone develops clinical disease, we find that in some of the relatives of type 1s, they also have a smaller pancreas. So there's this aspect of type 1 diabetes that is immune in nature, but there's also an aspect that may involve in just a smaller starting point of mass for the overall pancreas. And we now see some histological features of the disease that suggest that this is a disease of the whole pancreas, not just this small number of cells that happen to make insulin. So the pancreas does two things for you, really. It produces digestive enzymes to help you digest your food, but it also has these small clusters of cells that represent what we refer to as islets that have beta cells that make insulin. And it's really the loss of insulin that causes the profound disease presentation that we see in kids, where they essentially have blood glucose levels in the 500 to 600 range. So we're now doing imaging studies of individuals, living individuals, to understand when this loss of mass occurs, and using our high parameter or genetic studies, trying to understand what genetic elements throughout the genome may be contributing to this lower mass of the pancreas as well. So I have a very talented, she was originally a graduate student, now a postdoctoral fellow, who's also been working on understanding a type of protein that we refer to as insulin-like growth factors that are responsible for tissue homeostasis and growth throughout the entire body. And what she has found is that when we look in the circulation of individuals with type 1 diabetes, we actually see a profound reduction in both insulin-like growth factor 1 and 2. And that extends not only in individuals that have new onset disease, but also in individuals that we know are at high risk for developing disease. And there are antibodies that we know are predictive of individuals who will go on to develop disease. And as I mentioned, we can see a reduction in IGF levels in those individuals as well. So part of what we're doing overall is really trying to understand, can we predict individuals who are going to go on to develop disease so that we can intervene before their beta cells are destroyed. All right, so my laboratory runs a program that we refer to as HANDLE, and it's an acronym, and it stands for the Human Atlas for Neonatal and Developmental Early Life. This complex acronym that we like to make up all these titles. But what you're seeing here are some of our other efforts to understand how your immune system develops over time. So there's a couple key sites in your body that are responsible for your immune system developing. The first is really the bone marrow and the fetal liver. And then for T cells, you have this organ called the thymus, which sits almost right around above and in front of your heart region. And from these tissues, we can actually extract cells and look at all of the cells that are included within that tissue. And what you're looking at here, all of these pretty different colors that you're looking at represent individual single cells. So we have the technology now to understand all of the genes that are expressed at a single cell resolution. So this has dramatically changed our capacity to understand not only the different cells that make up an organ, but all of the genes that they express that makes them unique in terms of their function. So each one of these different colored dots represents a unique cell type. So we can get an amazing amount of information from a very small drop of blood by using this new technology in this process of single cell RNA sequencing. So RNA is essentially the genes that are involved in driving the function of all of the cells within your body. We also have new technologies that are coming on board to understand what tissues look like. So we can now label antibodies up to 40 different markers at a time in a tissue and understand what tissues look like using all of these different parameters. And this is just showing some of what the examples of what this looks like. So each one of these different colors represents a different cell type in the thymus itself. So there are these regions of the thymus that are chewing up dead and dying T cells as they try to develop. There are some B cells in the thymus. This is the primary site of T cell development for your body. And it's very active when you're young. So a newborn baby, all the way out until about the first decade of life is when your T cells are really actively developing. And then your thymus undergoes this process that we refer to as thymic involution. And what that really means is that these actively developing thymocytes start to slow down in terms of their development. They go out into the periphery, out into the body, where they go to work and they protect you from viruses and bacteria and all kinds of pathogens. But the thymus itself begins to change in its structure. So it goes from this active organ into having much more fibrous tissue, much more adipose tissue. So it goes through this process we refer to as thymic involution. All right, so I'm going to go through a quick concept now of where we're going in our laboratory. So one of the challenges that we've had in the field of type 1 diabetes is that we have good markers for disease development looking for antibodies, but we've never had a way of looking at T cells as a biomarker of disease progression. And there have been some challenging reasons for why that is the case. But you all have most likely heard of all of the hype around this notion of artificial intelligence. And we're now applying, so the University of Florida has the world's most powerful supercomputer at a public institution now. And it rivals, so it's the 20th, I think, most powerful supercomputer in the world. It rivals Microsoft's, some of Microsoft's supercomputers as well. And we're using the power. So to put it in context, it essentially has the power of several hundred very high performance personal computers. And we can utilize all of that computing power to look for biomarkers of disease progression in individuals that have type 1 diabetes. And the best representative example of that is that we can now sequence T cell receptors in peripheral blood. And what we're now trying to do is develop T cell receptor biomarkers of individuals that develop diseases like type 1 diabetes. And the best representative example of that is they basically drew blood from individuals that had a chronic viral infection, this CMV virus, and they could, with very high accuracy, predict if someone ever was exposed to this virus in their history. And that's an incredibly powerful idea, is that you could draw someone's blood and tell what they have been exposed to by sequencing their adaptive immune system. And so what that means is that, we can now essentially make very accurate predictions of whether or not someone has ever been exposed to CMV, COVID-19, but then also, are they at risk for these types of autoimmune diseases as well? So what this has allowed us to do is to really create a profile of type 1 diabetes in the immune system and make very accurate predictions about whether or not someone has some of these T cells that recognize things like insulin in the pancreas that go on to cause disease itself. What one of the key questions that I've mentioned already is that we really want to understand how to intervene in this disease process. And we've been using this drug, anti-thymocyte globulin, in patients that develop type 1 diabetes. And we can now utilize our capacity to look at all of the individual cells that we can find in peripheral blood by this single cell RNA sequencing technology. And we can identify what is the gene signature and what changes in the immune system in individuals that respond to a certain drug versus those that don't. So it's an incredibly powerful idea that you could take a very small amount of blood from an individual and understand what makes them a good responder to this drug versus a different drug. So we've published a number of papers on this, and we're finding that this population of T cells that we refer to as follicular helper T cells is reduced in individuals that are good responders to this drug ATG. So it's giving us really novel insight for these questions that in the past we've really struggled to understand. Why are some of these individuals good drug responders and others not? So the idea that I'm showing here is that from a very small blood draw, we can now sequence an individual's T cell receptors and understand what they are at risk for, what they've been exposed to in the past, and also, perhaps how they're going to respond to very different drugs. So here's just showing some of the T cell receptor sequences that are associated with risk for type 1 diabetes or cytomegalovirus or Epstein-Barr virus or HIV and many other sort of diseases and complications. So it's really, I think, going to transform a lot of the way that we understand how our immune systems work and what is going to make someone a good responder to drug X versus Y. So what we're hopefully moving towards is this idea of precision medicine. And what I mean by that is that if an individual goes into the clinic, a physician is going to take some portion, a small amount of blood, and try and understand what makes that person's disease unique. And what that will allow us to do is to tailor treatments to different individuals that we know are going to be better responders to drug X versus drug Y, to make more intelligent decisions in our patients so that they're not receiving drugs and side effects from drugs that are not going to work. All right. And then hopefully as well, when we treat with something like ATG, we'll be able to see the response profile in individuals to see if that drug is indeed working over time. All right, so I'll talk a little bit about, how do we speed things up? I always, one of the questions that always comes up in the field of research and clinical intervention is how do we improve the time from basic science research discoveries to effective therapies to treat patients with type 1 diabetes? And one of the ways that we're trying to speed this process up is to create these model systems, what we refer to as microphysiological systems, where we can put islets into these chips and then flow cells and different drugs over islets to see if we can model the disease process in a much faster manner. So I sit here on the 5th floor of the biomedical sciences building where we are studying type 1 diabetes, but just a couple floors down from me is a whole division of biomedical engineering. And the beauty of being in the same building is that we interact as a group. So all of these very talented engineers help us build these systems to understand how this autoimmune process is occurring. So we can now put islets into these microphysiological systems, these islets on a chip. We can put cells of the immune system in, and we can ask questions about, can we block this autoimmune process and model it in a way that'll speed up drug discovery for individuals in the clinic. So this is what some of that looks like in real time. And the beauty of this is that we can put this right up to a microscope, and we can actually image these islets being killed by cells of the immune system in real time. And I'll show what that looks like as well. So with some of my biomaterial engineering colleagues, this blue or green dot that you can see in the middle here is a cluster of insulin producing islet cells. And these red dots that you can see are actually T cells of your immune system. So they can actually, we can print them into these chips, and you can see these red cells swimming around these islets that will eventually be targeted and killed by these T cells. So basically what, for anyone to really understand what we're doing here, have you all used Purell hand sanitizer and kind of have an idea of what that feels like when you put it onto your hand? That's essentially the consistency of the stuff that we are working with here. It's very soft. We refer to this as soft matter. And what that allows us to do is to take a very small needle and print living cells in this gel material in a very defined way. So we can create these complex structures and model this disease process using these three-dimensional tissue culture systems. Another way of looking at this process, in this case, we have these target beta cells labeled with this red dye, and you can see some of them throughout this image. And then the killers, these T cells of the immune system, are in green. And what happens is that they kind of act like a swarm of bees, and they go in, they recognize these beta cells, and they can actually just kill them in real time. And you can see all of these red cells essentially dissolving in real time. So we can now have a way to kind of engineer these cells to make them and model this whole disease process in vitro. The other key thing that my lab is doing is we can use genetic engineering to change the specificity of T cells. And I know there's a lot of complex jargon there, but the basic idea is that the key process in type 1 diabetes is that you have a very small number of cells that are causing the disease process that recognize these insulin producing beta cells and are killing them. But your immune system is incredibly complex. So whenever you have something that kills something, you also have a population of cells that is there trying to keep your immune system in check. These cells called regulatory T cells. So we have the capacity to take these good cells, these regulatory cells, and try and direct them to the pancreas as well to block this autoimmune process. So one of the ways that we do that is by changing the receptor on the surface of the T cell itself to direct it to the pancreas to just shut down the autoimmune response. Because what we don't want to do is take an individual, a very young individual that has type 1 diabetes, and put them on a drug that is going to suppress their immune system for the rest of their life. That's not our goal. Our goal is really to keep their immune system largely in check, but to shut down just these T cells that are destroying these insulin producing beta cells. And we're now using the lessons that we've learned from cancer immunotherapy, and we're reversing the equation. So rather than targeting tumors and killing off tumors, we're putting these receptors onto these regulatory cells that keep the immune system in check and having them directed directly to the pancreas. All right, so I know there was a lot of material there, but I hope you get a sense of some of the overall goals of our program. It's really about understanding what is it that puts someone at increased risk for disease. Can we find what we refer to as biomarkers, so signals in their immune system that identifies individuals that are at increased risk for disease? And then can we intervene in that process and block some of the autoimmune response that is occurring? So with that, I'm happy to take any questions that have arisen through the course of some of those discussions, and I'll happily go back as well for if something wasn't clear along the way.
Okay, well, thank you very much for that fascinating lecture. I think just in the short time that I've been retired from the university, I was at that point keeping up with, what was going on in the diabetes research field. It's just amazing some of the new technologies now that you and Mark and your whole group have developed, and it's very, very exciting. So I wondered, so as far as like ATG and some of the drug approaches, so what class of drugs are these that we're really looking at, and how exactly would they work?
Yeah, so ATG is an old drug. It's been used for decades, but it's primarily been used for transplantation tolerance. And what we found was that if you give a much lower dose of it, about a third of the dose that you would normally give for individuals that are getting tissue transplants, it does a much better job at correcting the autoimmune response in the individuals that develop autoimmune diseases like type 1 diabetes. And one of the reasons that we think it does a much better job at that lower dose is that it preserves those good cops of the immune system, the regulatory T cells. So it's in a class of drugs that are generally referred to as biologics, because these are natural sort of proteins based on antibodies, as opposed to classic small molecule type drugs.
Okay, and is this so with what you're doing at OneVax, is this the area that you're working in, where you have those drugs encapsulated, and that's your drug delivery system? Oops. I think
idea that we could create a vaccine that would prevent someone from ever developing disease in the first place. So it would be essentially a process where we would identify someone that would be at higher genetic risk, or someone that had some of those antibody signatures that they might go on to develop disease. And we could give them a very low risk injection of a drug that, rather than boosting the immune system the way that you would want to do for something like COVID-19, we shut off the immune system. So we have drugs in there that selectively limit the immune response to something like insulin, essentially. And the idea is that it's based on biomaterial delivery of drug. So the best way I can describe our approach is that We use a drug called PLGA, which is the stuff that dissolvable sutures are made out of. So it's been used in the clinic quite a bit, and eventually it slowly releases drugs in the body over time. And we can create these with different drugs included inside that material itself, so that as that microparticle dissolves, it slowly releases those components. So it's a slightly different concept. ATG has been tried in individuals at disease onset, essentially. And what we're trying to do in OneVax is move back in the disease process into that prior to disease risk, so that we can prevent the disease from occurring in the first place.
Okay, I think we've got a few questions. Let's see if it's from the chat or Julianne, you've
Yeah, I
mentioned.
saw them raising their hands. Laverne
Okay.
Petzold.
So Laverne Petzold and can go first, and then Bev Winters.
We see you, Miss Winters. We'll take Laverne first. Unmute, Miss Petzold.
Laverne, you're muted. Okay.
Bev Winters, if you want to go ahead, and we'll get Laverne unmuted.
Okay, you're on.
Yes, I have a daughter.
Go ahead.
She has very crippling not osteo, but
Miss Winters, if you can speak a little closer to your microphone.
okay. My daughter lives in St. Pete. She has crippling psoriatic arthritis. They're waiting until she has both vaccines before they start her on some really heavy medicine. And I'm wondering, could they send a sample up here to use for the autoimmune sequencing? Or is there any place in St. Pete that would do that?
Yeah, so autoimmunity is a very broad term. So it encompasses, there are almost 80 different autoimmune conditions in total. So there's type 1 diabetes, but there's lupus, there's rheumatoid arthritis, multiple sclerosis, vitiligo. What your daughter has falls into that category of what we refer to as autoimmune diseases as well. And I understand the desire. What you don't want to do is, obviously you want their immune system fully intact so that they can mount a productive immune response to the COVID vaccine. And then I imagine after they have productive antibodies, protective antibodies, then they can begin to treat some of the autoimmunity in that case. I want to give a little bit of caution. So what we're doing for immune sequencing, unfortunately, is very much restricted right now to type 1 diabetes. But the company and that we've been working with to develop it very much is in the process of developing these biomarkers for other autoimmune conditions as well. This is very much an emerging field, though, so it's still a little bit early to bring it to any basic clinic just yet. What I will say is that the company that I work with is based in Seattle. It's called Adaptive Biotechnologies, and they're really one of the first companies to have an FDA-approved test to detect T cells to COVID-19. So they can very, very accurately predict if someone has ever received or ever had a prior infection with COVID-19. And obviously we're working carefully with them now to develop a test for type 1 diabetes, but it's still very much, unfortunately, in the research and development space right now. But hopefully over the next couple of years, we'll advance it into a clinical test that could be ordered at a classic sort of clinical setting.
All right, Miss Petzold, go ahead.
Am I unmuted now?
Yes.
Okay, good. My daughter developed some kind of a echo. Is there an echo? My
That's better.
daughter,
You're okay. I turned off Mr. Petzold's.
okay. My daughter developed type 1 diabetes at age 14. Two of her female classmates in grades 1 through 4 also developed type 1 diabetes around the same time that my daughter did. We always thought, is it a coincidence? They were classmates, or is there some kind of virus that could have precipitated this? Is that possible?
Yeah, absolutely. So there's been a lot of research about this idea that type 1 diabetes is linked to viral infections.
Mhm.
And I think for a long time, the field may have made the mistake of looking for viruses that directly infect beta cells and might kill beta cells. And I think what we now understand is that what happens when you get a viral infection is that your body makes a lot of inflammatory markers in response to that. And that can cause a break in immune regulation. So essentially one of the key things that you want to do when you have a viral infection is you want to clear that virus, and you want your T cells to get ramped up and geared up to mount a productive immune response. So what happens is that viral infections tend to follow with the onset of autoimmune conditions, but it doesn't necessarily mean that the virus is directly killing these insulin producing beta cells themselves. It might be just that the last chink in the sort of armor of how your immune system should be acting in someone that's at higher risk for developing the disease.
Thank you.
Okay, and just to follow up on that, so somebody that's genetically predisposed, so the DR3, DR4.
Yep, exactly.
Sort of thing.
Yeah.
Okay.
Absolutely.
Yeah.
And what we're seeing is actually, there's also been this, I think what has largely been a false notion in the past, that individuals that have autoimmunity have this overly robust immune response. And what we're seeing now in these very young trials with kids, where we track their blood over time, every 6 months to a year, is that when they get a viral infection, sometimes that viral infection actually persists and it doesn't clear effectively. So they might have, it's a little bit counterintuitive, but they actually may have a lower amount of immune regulation and activation than they should. And some of that may be due to some of those genetic susceptibility elements in the whole genome. So we see persistent viral infection in individuals that develop these antibodies and go on to develop disease later on, as opposed to, lots of kids get enteroviral infections. If your kids have ever been in daycare, you know that they come home and they have diarrhea, but it typically clears very quickly. In individuals that develop type 1 diabetes, for whatever reason, they have this long-term shedding of virus in their gut, and this persistent viral infection that seems to be a problem and be damaging their immune system.
Let me see. So I don't see any other questions in chat. I did have one question. So when you were looking at the whole pancreas itself, and it was discovered that the pancreas and T1D is but half the weight essentially. So which highlights really it's a disease of the entire pancreas, not just the insulin producing cells, although obviously that's probably the most profound outcome of that. But so as far as the exocrine portion with digestive enzymes, is that something that you uniformly see in people with type 1 diabetes? So loss of, specific digestive enzymes? Yeah,
that's a great question. And just this month, we published 2 papers in the journal Diabetes that individuals that have type 1 actually have a reduction in amylase, these digestive enzymes, lipase. So we see this even in circulation, that they have a reduction in what we would expect to be a normal pancreas function. It doesn't necessarily dramatically interfere with their ability to digest food in a normal way, but it is for us a very important biomarker of the disease process. And we see it in that when individuals that are at risk for disease, so we're looking to that as a surrogate, as a way of understanding who may actually go on to develop type 1 diabetes. The other thing that we've been doing is using ultrasound and MRI imaging of the pancreas to understand when that loss is occurring over the disease course. But there's a significant amount of just fibrosis within the entire pancreas itself. We see some scarring throughout the entire pancreas.
Well, I wonder if long-term though, because so much of our immune system is in the gut, right? So long-term, if your digestion is somewhat impaired, it's not that you can't digest food, but you're digesting it maybe incompletely, so that you now get these incompletely digested food particles in the small gut. And then we start seeing, we're learning a lot more about the so-called leaky gut and the effects of that on your immune system. And I'm just wondering if over time that you're sort of exacerbating your predisposition for autoimmune disease. So, over a decade of kind of not completely digesting food, if you're now setting yourself set up for new autoimmune diseases.
Yeah, we see a couple indicators of exactly what you mentioned. So the microbiome in individuals that have type 1 diabetes is different. Other gastrointestinal diseases track closely with type 1 diabetes. So about 13% of type 1s also have some symptoms of celiac disease. So these things do tend to co-occur. And there's obviously that link to enteroviral infections as well.
Paula,
Yes.
I do have a question that came to me. It's a multi-part question, if you don't mind.
Sure.
Can you describe the software used by UF Supercomputer? Is it related to the DNA modeling software recently reported as protein folding software? Who are the cooperating research organizations? And what is the promise of using these emerging computer tools?
Yeah, so the supercomputer was a gift, a $70 million gift by an individual that in collaboration with this software company or this hardware company called NVIDIA that provided the infrastructure for the supercomputer. And the software that runs on it is really based on user input. Oftentimes, when you talk about machine learning, that is a very broad category of things. Basically, what you're doing is you're asking computers to find patterns in data. So that data can have very different things. I mean, you show it 1 million images of a dog and you show it a new image and you say, is this a dog or is it a cat? And it will very rapidly recognize all of the patterns that are associated with it being a dog, and it'll make a very accurate call on that. So it can be images, it could be words in text, it could be very complex things like what we're doing to understand the immune system in sequencing DNA sequences and looking for patterns there. So all it really is it's a very powerful platform that can handle terabytes and petabytes of data and find patterns that humans could never find. That's essentially what, there's lots of complex terminology in associated with machine learning and artificial intelligence. But all it really is it comes down to it being a computer system where you feed in data and then you have a computer make connections in that data that you wouldn't normally be able to make yourself by looking at it. So I really enjoyed this, and unfortunately I have a back-to-back Skype call, so I have to take off, but it was a pleasure to meet with you all.